Chapter 11 Photosynthesis in higher plants. All animals, including human beings, depend on plants for their food. Have you ever wondered from where plants get their food? Green plants, in fact, have to make or rather synthesize the food they need, and all other organisms depend on them for their needs. The green plant make or rather synthesize the food they need through photosynthesis and are therefore called autotrophs. You have already learned that the autotrophic nutrition is found only in plants, and all other organisms that depend on the green plants for food are heterotrophs. Green plants carry out photosynthesis, a physiochemical process by which they use light energy to derive the synthesis of organic compounds. Ultimately, all living forms on earth depend on sunlight for energy. The use of energy from sunlight by plant doing photosynthesis is the basis of life on earth. Photosynthesis is important due to two reasons. It is the primary source of all food on earth. It is also responsible for the release of oxygen into the atmosphere by green plants. Have you ever thought what would happen if there were no oxygen to breathe? This chapter focuses on the structure of the photosynthetic machinery and the various reactions that transform light energy into chemical energy. 11.1 What do we know? Let us try to find out what we already know about photosynthesis. Some simple experiments you may have done in the earlier class have shown that chlorophyll, green pigment of the leaf, light, and carbon dioxide are required for photosynthesis to occur. You may have carried out the experiment to look for starch formation in two leaves, a variegated leaf or a leaf that was partially covered with black paper and exposed to light. On testing these leaves for the presence of starch, it was clear that photosynthesis occurred only in the green parts of the leaves in the presence of light. Another experiment you may have carried out where a plant of a leaf is enclosed in a test tube containing some KOH soaked cotton which absorb carbon dioxide while the other half is exposed to air. This setup is then placed in light for some time. On testing for the presence of starch later into parts of the leaf, you must have found that the exposed part of the leaf tested positive for starch while the portion that was in the tube tested negative. This showed that carbon dioxide was required for photosynthesis. Can you explain how this conclusion could be drawn? 11.2 Early experiment. It is interesting to learn about those simple experiments that lead to a gradual development in our understanding of photosynthesis. Joseph Priestley (1733 to 1804) in 1770 performed a series of experiments that revealed the essential role of air in the growth of green plant. Priestley, you may recall, discovered oxygen in 1774. Priestley observed that a candle burning in a closed space, a bell jar soon gets extinguished. Figure 11.1a, b, c, d. Similarly, a mouse would soon suffocate in a closed space. He concluded that a burning candle or an animal that breathed in the air both somehow damaged the air, but when he placed a mint plant in the same bell jar, he found that the mouse stayed alive and the candle continued to burn. Priestley hypothesized as follows: plant restored the air, whatever breathing animals and burning candles removed. Can you imagine how Priestley would have conducted the experiment using a candle and a plant? Remember, he would need to rekindle the candle to test whether it burns after a few days. How many different ways can you think of to light the candle without disturbing the setup? Using a similar setup as the one used by Priestley but by placing it once in the dark and once in the sunlight. Jan Agnew (1730 to 1799) showed that sunlight is essential to the plant process that somehow purifies the air fouled by burning candles or breathe animals. Igneous is an elegant experiment with an aquatic plant showed that in bright sunlight, small bubbles were formed around the green parts while in the dark they did not. Later he identified these bubbles to be of oxygen. Hence he showed that it is only the green part of the plant that could release oxygen. It was not until about 1854 that Julius von Sachs provided evidence for production of glucose when plant grows. Glucose is usually stored as starch. His Later studies showed that the green substance in plant chlorophyll as we know it now is located in special bodies later called chloroplast. Within plant cell he found that the green parts in plant is where glucose is made and that the glucose is usually stored as starch. Now consider the interesting experiment done by T.W. Engelmann 1843 to 1909. Using a prism he split light into its spectral component and then illuminated a green alga cladophora placed in a suspension of aerobic bacteria. The bacteria were closed to detect the site of oxygen evolution. He observed that the bacteria accumulated mainly in the region of blue and red light of the split spectrum. A first action spectrum of photosynthesis was thus described. It resembles roughly the absorption of spectra of chlorophyll A and B discussed in section 11.4. By the middle of the 19th century, the key feature of plant photosynthesis we know were known, namely that plant could use light energy to make carbohydrates from carbon dioxide and water. The empirical equation representing the total process of photosynthesis for oxygen evolving organism was then understood as where CH2O represented a carbohydrate, example glucose, a 6 carbon sugar. A milestone contribution to the understanding of photosynthesis was that made by a microbiologist Cornelius Van Neel 1897 to 1985, who based on his studies of purple and green bacteria demonstrated that photosynthesis is essential, a light dependent reaction in which hydrogen from a suitable oxidizable compound reduce carbon dioxide to carbohydrates. This can be expressed by in green plant water is the hydrogen donor and is oxidized to O2. Some organisms do not release O2 during photosynthesis when H2S instead is the hydrogen donor for purple and green sulfur bacteria. The oxidation product is sulfur or sulfate depending on the organism and not oxygen. Hence, he inferred that the O2 evolved by the green plant comes from H2O, not from carbon dioxide. This was later proved by using radioisotopic technique. The correct equation that would represent the overall process of photosynthesis is therefore where C6H12O6 represents glucose, the O2 released is from water. This was proved using radioisotopes technique. Note that this is not a single reaction but description of a multi-step process called photosynthesis can explain why 12 molecules of water as substrate are used in the equation given below. 11.3 Where does photosynthesis take place? You would have of course answer in the green leaf or in the chloroplast based on what you earlier read in chapter 8. You are definitely right. Photosynthesis does take place in the green leaves of plant, but it does so also in other green parts of the plant. Can you name some other parts where you think photosynthesis may occur? You would recollect from previous until unit that the mesophyll cells in the leaves have a large number of chloroplasts. Usually the chloroplasts align themselves along the walls of the mesophyll cells such that they get the optimum quantity of the incident light. When do you think the chloroplast will be aligned with their flat surface parallel to the walls? When would they be perpendicular to the incident light? You have studied the structure of chloroplast in chapter 8. Within the chloroplast there is membranous system consisting of grana, the stroma, lamella and the matrix stroma. Figure 11.2. There is a clear division of labor within the chloroplast. The membrane system is responsible for trapping the light energy and also for the synthesis of ATP and NADPH. Stroma enzymatic reaction synthesis sugar which is done form starch. The former setup of reaction, since they are directly light driven, are called like reaction. Photochemical reaction, the later are not directly light driven but are dependent on the products of light reaction ATP and NADPH. Hence, to distinguish the later, they are called by convention as dark reaction, carbon reaction. However, this should not be constructed to mean that they occur in darkness or that they are not light dependent. 11.4 How many types of pigments are involved in photosynthesis? Looking at plant, have you ever wondered why and how there are so many shades of green in their leaves? Even in the same plant, we can look for an answer to this question by trying to separate the leaf pigment of any green plant through paper chromatography. A chromatographic separation of the leaf pigment shows that the color that we see in leaves is not due to the single pigment but due to four pigments: chlorophyll A, bright or blue green in the chromatogram, chlorophyll B, yellow green, xanthophyll yellow, and carotenoid yellow to yellow orange. Let us now see what roles various pigments play in photosynthesis. Pigments are substances that have an ability to absorb light at specific wavelength. Can you guess which is the most abundant plant pigment in the world? Let us study the graph showing the ability of chlorophyll A pigment to absorb light of different wavelengths. Figure 11.3a. Of course, you are familiar with the wavelength of the visible spectrum of light as well as the Vibgyor. From figure 11.3a, can you determine the wavelength color of light at which chlorophyll A shows the maximum absorption? Does it show another absorption peak at any other wavelengths too? If yes, which one? Now look at figure 11.3b showing the wavelength at which maximum photosynthesis occur in a plant. Can you see that the wavelength at which there is maximum absorption by chlorophyll A in the blue and the red region also shows higher rate of photosynthesis? Hence we can conclude that chlorophyll A is the chief pigment associated with photosynthesis. But by looking at figure 11.3c, can you say that there is complete one-to-one overlap between the absorption spectrum of chlorophyll A and the reaction spectrum of the photosynthesis? These graphs together show that the most of the photosynthesis take place in the blue and red region of the spectrum. Some photosynthesis does take place at the other wavelength of the visible spectrum. Let us see how this happens. Though chlorophyll is the major pigment responsible for trapping light, other thylakoid pigments like chlorophyll B, xanthophyll, and carotenoid, which are called accessory pigments, also absorb light and transfer the energy to chlorophyll A. Indeed, they not only enable to wider range of wavelength incoming light to be utilized for photosynthesis but also protect chlorophyll A from photooxidation. 11.5 What is light reaction? Light reaction or the photochemical phase include light absorption, water splitting, oxygen release, and the formation of high energy chemical intermediates. ATP and NADPH, several protein complexes are involved in the process. The pigments are organized into two discrete photochemical light harvesting complexes, LHC, within the photosystem I, PS1, and photosystem second, PS2. These are named in the sequence of their discovery and not in the sequence in which they function during the light reaction. The LHC are made up of hundreds of pigments molecules bound to protein. Each photosystem has all the pigment except one molecule to chlorophyll A, forming a light harvesting system also called antenna. Figure 11.4 These pigments help to make photosynthesis more efficient by absorbing different wavelengths of light. The single chlorophyll A molecule forms the reaction center. The reaction center is different in both the photosystems. In PS1, the reaction center chlorophyll A has an absorption peak at 700 nanometer, hence is called P700, while in PS second it has the absorption maximum at 680 nm and it is called P680. 11.6 The elegant transport. In photosystem second, the reaction center chlorophyll A absorbs 680 nanometer wavelength of red light, causing electron to become excited and jump into an orbit further from the atomic nucleus. These electrons are picked up by an electron acceptor which passes them to an electron transport system consisting of cytochromes. Figure 11.5 This movement of electron is downhill. In terms of an oxidation reduction or redox potential scale, the electrons are not used up as they pass through the electron transport chain but are passed on to the pigments of photosynthesis PS1. Simultaneously, electrons in the reaction center of PS1 are also excited when they receive red light of wavelength 700 nanometer and are transferred to another acceptor molecule that has a greater redox potential. These electrons then are moved downhill again, this time to a molecule of energy rich NADP. The addition of these electrons reduce NADP to NADPH plus H positive. This whole scheme of transfer of electrons starting from the PS second uphill to the acceptor down to the electron transport chain to PS1, excitation of electrons transfer to another acceptor, and finally downhill to NADP, reducing it to NADPH plus H is called the Z scheme due to its characteristic shape. Figure 11.5. This shape is formed when all the carriers are placed in a sequence on the redox potential scale. 11.6.1 Splitting of water. You would then ask, how does PS second supply electrons continuously? The electrons that were moved from photosystem second must be replaced. This is achieved by electrons available due to splitting of water. The splitting of water is associated with the PS second. Water is split into 2H positive and electrons. This creates oxygen, one of the net products of photosynthesis. The electrons needed to replace these removed from photosystem first are provided by photosystem second. We need to emphasize here that the water splitting complex is associated with the PS second, which itself is physically located on the inner side of the membrane of the thylakoid. Then where are the protons and O2 formed, likely to be released in the lumen or on the outer side of the membrane. 11.6.2 Cyclic and non-cyclic photophosphorylation. Living organisms have the capability of extracting energy from oxidable substance and store this in the form of bond energy. Special substances like ATP carry this energy in their chemical bond. This process through which ATP is synthesized by cells in mitochondria and chloroplast is named phosphorylation. Photophosphorylation is the synthesis of ATP from ADP and inorganic phosphate in the presence of light. When the two photosystems work in a series, first PS second and then the PS first, a process called non-cyclic photophosphorylation occur. The two photosystems are connected through an electron transport chain as seen earlier in the Z scheme. Both ATP and NADPH plus H are synthesized by this kind of electron flow. Figure 11.5. When only PS1 is functional, the electron is circulated within the photosystem and the phosphorylation occurs due to the cyclic flow of electron. Figure 11.6. A possible location where this could be happening in the stroma lamella, while the membrane or lamella of the grana have both PS1 and PS second. The stroma lamella membrane lack PS2 as well as the NADP reductase enzyme. The excited electron does not pass on to the NADP but is cycled back to the PS1 complex through the electron transport chain. Figure 11.6. Cyclic flow hence result only in the synthesis of ATP but not of NADPH plus H. Cyclic phosphorylation occur when the only light of wavelength beyond 680 nanometer are available for excitation. 11.6.3 Chemosomatic hypothesis. Let us now try and understand how actually ATP is synthesized in the chloroplast. The chemosomatic hypothesis has been put forward to explain the mechanism. Like in respiration and photosynthesis too, ATP synthesis is linked to development of a proton gradient across a membrane. This time these are the membrane of thylakoid. There is one difference through here. The proton accumulation is toward the inside of the membrane in the lumen. In respiration, protons accumulate in the intermembrane space in the mitochondria when electrons move through the EDS. Chapter 12. Let us understand what caused the proton gradient across the membrane. We need to consider again the process that take place during the activation of electron and their transport to determine the steps that cause a proton gradient to develop. Figure 11.7. Since splitting of the water molecules take place on the inner side of the membrane, the protons or hydrogen ions that are produced by the splitting of water accumulate within the lumen of the thylakoid. B. As electrons move through the photosystem, protons are transported across the membrane. This happens because the primary acceptor or the proton, which is located towards the outer side of the membrane, transfers its electron not to an electron carrier but to an hydrogen carrier. Hence, this molecule removes a proton from the stroma while transporting an electron. when this molecule passes on its electron to the electron carrier on the inner side of the membrane, the proton is released into the inner side of the lumen side of the membrane. C. The NADP reductase enzyme is located on the stroma side of the membrane along with electrons that come from the acceptor of electrons of PS1. Protons are necessary for the reduction of NADP to NADPH plus H. These protons are also removed from the stroma. Hence, within the chloroplast, protons in the stroma decrease in number, while in the lumen there is accumulation of protons. This creates a proton gradient across the thylakoid membrane as well as the measurable decrease in pH in the lumen. Why are we so interested in the proton gradient? This gradient is important because it is breakdown of this gradient that leads to the synthesis of ATP. This gradient is broken down due to the movement of protons across the membrane to the stroma through the transmembrane channels of the CF0 of the ATP synthesis. The ATP synthesis enzyme consists of two parts. One called the CF0 is embedded in the thylakoid membrane and forms a transmembrane channel that carries out facilitated diffusion of protons across the membrane. The other portion is called the CF1 and protrudes on the outer side of the thylakoid membrane on the side that faces the stroma. The breakdown of the gradient provides enough energy to cause a conformational change in the CF1 particle of the ATP synthesis, which make the enzyme synthesize several molecules of energy-packed ATPs. Chemosynthesis require a membrane, a proton pump, a proton gradient, and ATP synthesis. Energy is used to pump proton across a membrane to create a gradient or a high concentration of proton. Within the thylakoid lumen, ATP synthesis has a channel that allows diffusion of proton back across the membrane. This releases enough energy to activate an ATP synthesis, an enzyme that catalyzes the formation of ATP along with the NADPH produced by the movement of electron. The ATP will be used immediately in the biosynthetic reaction taking place in the stroma, responsible for fixing carbon dioxide and synthesis of sugar. 11.7 Where are the ATP and NADPH used? We learned that the products of the light energy are ATP, NADPH, and oxygen. Or of these, oxygen diffuse out of the chloroplast, while ATP and NADPH are used to derive the process leading to the synthesis of food. More accurately, sugar. This is the biosynthetic phase of the photosynthesis. This process does not directly depend on the presence of light, but is depend on the products of the light reaction, ATP and NADPH, beside carbon dioxide and water. You may wonder how this could be verified. It is simple. Immediately after light become unavailable, the biosynthetic process continuously for some time, then stops. If then light is made available, the synthesis start again. Can we hence say that calling the biosynthetic phase as the dark reaction is a misnomer? Discuss this amongst yourself. Let us now see how the ATP and NADPH are used in the biosynthetic phase. We saw earlier that carbon dioxide is combined with water to produce CH2ON or sugar. It was of interest to scientists to find out how this reaction proceed, or rather what was the first product formed when carbon dioxide is taken into a reaction or fixed. Just afterward was second among the several efforts to put radioisotopes to beneficial use. The work of Melvin Kelvin is exemplary. The use of radioactive C14 by him is in algal photosynthesis study lead to the discovery that the first carbon dioxide fixation product was a 3-carbon organic acid. He also contributed to working out the complete biosynthetic pathway, hence it was called the Kelvin cycle. After him, the first product identified was 3-phosphoglyceric acid, or in short PGA. How many carbon atoms does it have? Scientists also tried to know whether all plants have PGA as the first product of carbon dioxide fixation, or whether any other product was formed in other plants. Experiments conducted over a wide range of plants lead to the discovery of another group of plants where the first stable product of carbon dioxide fixation was again an organic acid, but one which had 4 carbon atoms in it. This acid was identified to be oxalic acid, or OAA. Since then, carbon dioxide assimilation during photosynthesis was said to be of 2 main types. Those plants in which the first product of carbon dioxide fixation is a C3 acid, the C3 pathway and those in which the first product was C4 acid. The C4 pathway, these two groups of plants showed other associated characteristics that we will discuss later. 11.7.1 The primary acceptor of carbon dioxide. Let us now ask ourselves a question that we asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which, after accepting facing carbon dioxide, would have 3 carbon of PGA? The studies were unexpectedly showed that the acceptor molecules was a 5 carbon ketose sugar ribulose biphosphate RUBP. Did any of you think of this possibility? Do not worry, the scientists also took a long time to conduct many experiments to reach this conclusion. They also believed that since the first product was a C3 acid, the primary acceptor would be a 2 carbon compound. They spent many years trying to identify a 2 carbon compound before they discovered the 5 carbon RUBP. 11.7.2 The Calvin cycle. Calvin and his co-workers then worked out the whole pathway, ensured that the pathway operated in a cyclic manner. The RUBP was regenerated. Let us now see how the Calvin pathway operates and where the sugar is synthesized. Let us at the outset understanding very clearly that the Calvin pathway occurs in all photosynthetic plants. It does not matter whether they have C3 or C4 or any other pathway. Figure 11.8 For ease of understanding, the Calvin cycle can be described under 3 stages: carboxylation, reduction, and regeneration. First, carboxylation. Carboxylation is the fixation of carbon dioxide into a stable organic intermediate. Carboxylation is the most crucial step of the Calvin cycle, where carbon dioxide is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which result in the formation of 2 molecules of 3 PGA. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase, oxygenase, or Rubisco. Second, reduction. These are a series of reactions that lead to a formation of glucose. This step involves utilization of 2 molecules of ATP for phosphorylation and 2 of NADPH for reduction. Per carbon dioxide molecule fixed, the fixation of 6 molecules of carbon dioxide and 6 turns of the cycle are required for the formation of 1 molecule of glucose from the pathway. Regeneration of the carbon dioxide acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration step requires 1 ATP for phosphorylation to form the RUBP. Hence, for every carbon dioxide molecule entering the Calvin cycle, 3 molecules of ATP and 2 NADPH are required. It is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. To make 1 molecule of glucose, 6 turns of the cycle are required. Work out how many ATP and NADPH molecule will required to make 1 molecule of glucose through the Calvin pathway. It might help you to understand all of this if we look at what goes in and what comes out of the Calvin cycle. 11.8 The C4 pathway. Plants that are adapted to dry tropical region have the C4 pathway mentioned earlier. Through these plants have the C4 oxalic acid as the first carbon dioxide fixation product. They use the C3 pathway or the Calvin cycle as the main biosynthetic pathway. Then in what way they different from C3 plant? This is a question that you may reasonably C4 plants are special. They have some special type of leaf anatomy. They tolerate higher temperature. They show a response to high light intensities. They lack a process called photorespiration and have greater productivity of biomass. Let us understand these one by one. Study vertical section of leaves, one of a C3 plant and the other of a C4 plant. Do you notice the difference? Do both have the same types of mesophyll? Do they have similar cell around the vascular bundle sheath? The particularly large cell around the vascular bundles of the C4 plant are called bundle sheath cell, and the leaves which have such anatomy are called the Kranz anatomy. Kranz means breadth and is a reflection of the arrangement of leaves. A bundle sheath cell may have formed several layers around the vascular bundles. They are characterized by having a large number of chloroplasts, thick wall imprevision to gaseous exchange, and no intercellary spaces. You may like to cut a section of the leaves of C4 plant maize or sorghum to observe the crown's anatomy and the distribution of the mesophyll cell. It would be interesting for you to collect leaves of diverse species of the plant around you and cut vertical section of the leaves. Observe under the microscope. Look for the bundle sheath around the vascular bundle. The presence of the bundle sheath would help you to identify the C4 pathway. Now study the pathway shown in figure 11.9. This pathway that has been named the H and Slack pathway is again a cyclic process. Let us study the pathway by listing the steps. Primary carbon dioxide acceptor is a 3-carbon molecule phosphenol pyruvate PEP is present in the mesophyll cell. The enzyme responsible for the fixation of PEP carboxylase or peptidase. It is important to register that the mesophyll cell lack Rubisco enzyme. The C4 pathway OAA is formed in the mesophyll cell. It then forms other 4-carbon compounds like malic acid, aspartic acid in the mesophyll cell itself, which are transported to the bundle sheath cell. In the bundle sheath cell, these C4 acids are broken down to release carbon dioxide and a 3-carbon molecule. The 3-carbon molecule is transported back to the mesophyll, whether it is converted to PEP again, thus completing the cycle. The carbon dioxide release the bundle sheath cell enter the C3 or the Kelvin pathway, a pathway common to all the plant. The bundle sheath cell are rich in an enzymatic ribulose biphosphate carboxylase enzyme oxygenase Rubisco but lack peptidase. Thus, the basic pathway that result in the formation of the sugar. The Kelvin pathway is common to the C3 and C4 plant. Did you note that the Kelvin pathway occur in all the mesophyll cell of the C3 plant? In the C4 plant, it does not take place in the mesophyll cell, but it does so only in the bundle sheath cell. 11.9 Photorespiration. Let us try and understand one more process that creates an important difference between C3 and C4 pathway. C4 plant photorespiration. To understand photorespiration, we have know a little bit more about the first step of the Kelvin pathway. The first carbon dioxide fixation step is the reaction where RuBP combines with carbon dioxide to form two molecules of 3-PGA that is catalyzed by Rubisco. Rubisco, that is the most abundant enzyme in the world. Do you wonder why it's characterized by the fact that its active site can bind to both carbon dioxide and oxygen, hence the name? Can you think how this could be possible? Rubisco has much greater affinity for carbon dioxide when the carbon dioxide ratio oxygen is nearly equal. Imagine what would happen if we were not so. This binding is competitive. It is the relative concentration of carbon dioxide and oxygen that determines which of the two will bind to the enzyme. In C3 plant, some oxygen does bind to Rubisco, hence carbon dioxide fixation decreased. Here the RuBP, instead of being converted to two molecules of PGA, binds with oxygen to form one molecule of phosphoglycerate and phosphoglycolate to carbon in a pathway called photorespiration. In the photorespiration pathway, there is neither synthesis of sugar nor of ATP, rather it result in the release of carbon dioxide with the utilization of ATP. In the respiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not known yet. In C4 plant, photorespiration does not occur. This is because they have a mechanism that increases the concentration of carbon dioxide at the enzyme site. This takes place when the C4 acid from the mesophyll is broken down the bundle sheath cell to release carbon dioxide. This results in increasing the intracellular concentration of carbon dioxide. In turn, this ensures that the Rubisco function as a carboxylase, minimizing the oxygenase activity. Now that you know that the C4 plant lack photorespiration, you probably can understand why productivity and yield are better in these plants. In addition, these plants show tolerance to higher temperature. Based on the above discussion, can you compare plants showing the C4 and C3 pathway? Use the table format given in the 11.1 and fill in the formation. Table 11.1, fill in the column 2 and column 3 in the table to highlight the difference between C3 and C4 pathway. 11.10, factors affecting photosynthesis. An understanding of the factors that affect photosynthesis is necessary. The rate of photosynthesis is very important in determining the yield of plant, including crop plant photosynthesis under the influence of several factors, both internal plant or external. The plant factor include the number, size, age, and orientation of leaf, mesophyll cell, and chloroplast, internal carbon dioxide concentration, and the amount of chlorophyll. The plant or internal factor are dependent on the genetic predisposition and the growth of the plant. The external factor would include the availability of sunlight, temperature, carbon dioxide concentration, and water. As a plant photosynthesis, all these factors will simultaneously affect its rate. Hence, through several factors interact and simultaneously affect photosynthesis or carbon dioxide fixation. Usually, one factor is the major cause or is the one that limits the rate. Hence, at any point, the rate will be determined by the factor available at suboptimal levels. When several factors affect any biochemical process, Blackman's 1905 law of limiting factor come into effect. This state the following: if a chemical process is affected by more than one factor, then the rate will be determined by the factor which is nearest to its minimal value. It is the value which directly affects the process if its quantity is changed. For example, despite the presence of a green leaf in optimal light and carbon dioxide condition, the plant may not photosynthesize. If the temperature is very low, this leaf, if given the optimal temperature, will start photosynthesizing. 11.10.1 Light. We need to distinguish between light quality, light intensity, and the duration of exposure of light. While discussing light as a factor that affects photosynthesis, there is a linear relationship between incident light and carbon dioxide fixation rates at low light intensities. At higher light intensity, gradually the rate does not show further increase as other factors become limiting. Figure 11.10. What is interesting to note is that light saturation occurs at 10% of the full sunlight, hence except for plants in shade or in dense forest, light is rarely a limiting factor in nature. Increase in incident light beyond a point cause the breakdown of chlorophyll and a decrease in photosynthesis. 11.10.2 Carbon dioxide concentration. Carbon dioxide is the major limiting factor for photosynthesis. The concentration of carbon dioxide is very low at the atmosphere, between 0.03 and 0.04%. Increase in the concentration up to 0.05% can cause an increase in the carbon dioxide fixation rates. Beyond this, the level can become damaging over longer period. The C3 and C4 plant respond differently to CO2 concentration at low light conditions. Neither group will respond to high carbon dioxide condition at high light intensities. Both C3 and C4 plants show increase in the rate of the photosynthesis. What is important to note is that the C4 plant shows saturation at about 360 ml, while C3 respond to increased carbon dioxide concentration and this saturation is seen only beyond 450 ml. Thus, current availability of carbon dioxide levels is limiting to the C3 plant. This fact that C3 plant respond to higher carbon dioxide concentration by showing increased rate of photosynthesis, leading to the higher productivity, has been used for some greenhouse crops such as tomatoes and bell pepper. They are allowed to grow in carbon dioxide and enriched atmosphere that leads to higher yield. 11.10.3 Temperature. The dark reaction being enzymatic are temperature controlled through the light reaction are also temperature sensitive. They are affected to a much lesser extent. C4 plant respond to higher temperature and show rather rate of synthesis, while C4 plant have much lower temperature optimum. The temperature optimum for photosynthesis of different plant also depend on the habitat that they are adapted to. Tropical plant have a higher temperature optimum than the plant adapted to tolerate temperate climates. 11.10.4 Water. Even though water is one of the most reactant in the light reaction, the effect of water as the factor is more through its effect on the plant rather than directly or photosynthesis. Water stress causes the stomata to close, hence reducing the CO2 availability. Beside, water stress also makes the leaf wilts, thus reducing the surface area of the leaves and the metabolic activity as well. Summary: green plants make their own food by photosynthesis. During the process, carbon dioxide from the atmosphere is taken by leaves through stomata and used for making carbohydrates, principally glucose and starch. Photosynthesis take place only in the green parts of the plant, mainly the leaves. Within the leaves, the mesophyll cell have a large number of chloroplasts that are responsible for carbon dioxide fixation. Within the chloroplast, the membranes are sites for the light reaction, while the chemosynthetic pathway occur in the stroma. Photosynthesis has two stages: the light reaction and the carbon fixing reaction. In the light reaction, the light energy is absorbed by the pigments present in the antenna and funneled to special chlorophyll A molecules called the reaction center chlorophyll. There are two photosystems, PS1 and PS2. PS1 has 700 nanometer absorbing chlorophyll A P700 molecule at its reaction center, while PS2 has 680 reaction center that absorb red light at 680 nanometer. After absorbing light, electrons are excited and transferred through PS2 to PS1 and finally to NAD, forming NADH. During this process, a proton gradient is created across the membrane of the thylakoid. The breakdown of the protons gradient due to movement through the F0 part of the ATP's enzyme release enough energy for the synthesis of ATP. Splitting of water molecules is associated with the PS2, resulting in the release of oxygen protons and transfer of the electron to PS2. In the carbon fixation cycle, carbon dioxide is added to the enzyme Rubisco to a 5-carbon compound RUPPA that is converted to 2 molecule of 3-carbon PGA. This is then converted to sugar by the Calvin cycle, and the RUBP is regenerated during the process. ATP and NADPH synthesized in the light reaction are utilized. Rubisco also catalyzes a wasteful oxygenation reaction in C3 plant photorespiration. Some tropical plants show a special type of photosynthesis called C4 pathway. In these plants, the first product of carbon dioxide fixation that takes place in the mesophyll is a 4-carbon compound in the bundle sheath cell as the Calvin pathway is carried out for the photosynthesis of carbon dioxide.